Assalamu alaikum. This video will discuss document 3 of chapter 5, sex-linked diseases. In the introduction to this chapter, you have recalled that genes could be autosomal genes carried by the autosomal chromosomes or sex-linked genes that are carried by the gonosomal chromosomes X and Y. Some sex-linked genes are responsible for serious genetic disorders like Duchenne muscular dystrophy, DMD, which is a disease characterized by the weakening of voluntary muscles, or rickets, which is another disease characterized by deformities in the skeleton. In this video, you're not going to study the progression of such diseases, but you are going to learn how to use the pedigree of families with such diseases to determine the location of their corresponding gene. But before we start, let's recall the chromosomal basis of sex determination. In other words, how is the gender of a human determined to be a male or a female? What you already know is that a female possesses in her cells two X chromosomes, while a male possesses an X and another Y chromosome. That's why, during gamete production, all gametes produced by a female will have the gonosome X, while male gametes are of two types with equal probabilities. Half of the male gametes possess X chromosome, while the other half possess Y chromosome. That's why, after fertilization, the possibility that the zygote is a female is 50%, which is equal to the possibility that the zygote is a male. Now, to understand the transmission of a sex-linked disease, let's consider a certain gene carried by the non-homologous part of X chromosome. Let's consider the normal allele of this gene to be dominant, while the mutant allele that causes the disease to be recessive. So in such case, a female that has two normal alleles carried by her two X chromosomes will be certainly healthy, not affected by the disease. The female that is heterozygous with only one mutant allele will also be healthy, given that the allele for the disease is masked by the dominant normal one. The only female affected by this disease will be the one having a mutant allele on each of her X chromosomes. But when it comes to a male, this allele is not carried on the Y chromosome and only carried on the non-homologous part of X chromosome. Thus, only one mutant allele on the X chromosome will make the male affected by the disease, since this allele is not masked by any other allele. And the male will only be healthy if the X chromosome carries the normal allele. So, based on the above example, do you think that both males and females are equally affected with a sex-linked disease? To help you answer this question, let's consider the following couple. Both parents are healthy, but the mother is heterozygous with one mutant allele. Let's check what will be the possible genotype of the children of this couple concerning this sex-linked disease. Using the Punnett square table, we can predict all possible genotypes of the children concerning the disease. You can obviously see that this couple can never give birth to an affected girl, because a girl will always inherit the normal allele carried on the X chromosome from her healthy father. We say that the risk for this couple to give birth to an affected girl is 0. But when it comes to boys, since boys take the Y chromosome from their father, then they will express the allele carried on the non-homologous part of X chromosome, whether it's normal or mutant. That is why this couple is at the risk of having an affected boy. Actually, the risk of this couple to have an affected boy is equal to quarter, and you will learn about how to calculate the risk in another video. But as you see, males and females are not equally affected by sex-linked diseases. Actually, males are most likely to be affected with such diseases, and this is one important characteristic to use when you want to prove a gene to be sex-linked. Let's consider this pedigree of a family with members being affected by the recessive disease Duchenne muscular dystrophy. The affected members are shown in red, while the healthy ones are shown in blue. Now, is there any clue in this pedigree that suggests that the gene responsible for the disease is sex-linked? If you are thinking about the fact that all affected members are males, you're absolutely correct. When in a pedigree you see only males being affected, this is an evidence that the gene under study is sex-linked for sure. Another evidence that you could use to prove that a certain disease is sex-linked is when you notice a difference in the gender of affected children accompanied with the difference in the gender of the affected parent. Let me make this simple. In this example about a dominant sex-linked disease, notice that when the father is the affected parent, all the daughters are affected. But when the mother is the affected parent, both boys and girls could be affected. This difference in the results suggests that the gene responsible for the disease is sex-linked, because if it was an autosomal gene, then it won't make any difference if the affected parent was the father or the mother. But when you are asked to determine the location of a gene depending on the pedigree analysis, it's not enough to say it's sex-linked. Actually, you should further analyze the pedigree to determine if the gene is located on the non-homologous part of X or the non-homologous part of Y chromosome. For the genes that are located on the non-homologous part of X, you should follow the strategy of elimination. It means that to prove the gene to be linked to the non-homologous part of X, eliminate the possibility that it's on the non-homologous part of Y. Let's go back to the example of DMD and try to locate the gene responsible for this disease. So after we already proved the gene for DMD to be sex-linked, let's take into consideration the affected male 3 of the second generation. If the allele for the disease is carried by the non-homologous part of Y, then the genotype of this male would be XYD. And since a male inherits his Y chromosome from his father, then the father should be also affected, which is not the case, obviously. Then the allele for DMD cannot be carried by the non-homologous part of Y, and thus it's carried by the non-homologous part of X. Now, when it comes to dominant sex-linked diseases such as rickets, you can use the same strategy to determine the location of the gene responsible for rickets. Given that the gene is sex-linked, if it was located on the non-homologous part of Y, then the affected male 1 of the third generation will have the genotype XYR, which will make his father also affected, but this is not the case since the father is healthy. Then, the gene for rickets is located on the non-homologous part of X. Now, when it comes to a gene linked to the non-homologous part of Y chromosome, you will be directly capable of recognizing its inheritance. This pedigree shows a family with males having the trait hairy ears. It's obvious that the gene responsible for this trait is sex-linked, since all members with this phenotype are males. With no exception, every affected male has an affected father, which suggests that the gene for this trait is linked to the non-homologous part of Y chromosome. So for now, using a pedigree analysis, you are capable of identifying sex-linked diseases and locate the gene responsible for the disease, whether on the non-homologous part of X or Y. In the next video, you will learn how to determine the location of autosomal genes, referring to pedigree analysis as well. Assalamu alaikum.